Useful factoids out onto the table. So your brain has two cerebral hemispheres that are each about 1,000 square centimeters. That's about a 13-inch pizza that's crumpled up to fit inside your skull. A macaque monkey, by comparison, is 1/10 the size, about the size of a chocolate chip cookie for each hemisphere. Each sheet is thin. Our cortex is only a little bit thicker than that of a macaque. One of the most striking characteristics is that the macaque has a highly stereotyped pattern of convolutions. The human, as we'll see, is much more variable in many regions of cortex. The number of cortical areas is a hugely important and intriguing question. By some measures, there are well over 100, perhaps 130 or so cortical areas in the macaque. In humans, it's been speculated for many years that there are more areas in humans than in the macaque, but until recently it's been very hard to get a good reading on that. From evidence that I will have only time to summarize the bottom line here, Matt Glasser, who will be talking later about other aspects of cortical organization, has now provided evidence for at least 178 cortical areas as analyzed from multimodal analysis of Human Connectome Project data. But this is really here for just the context that we have a lot of areas that, as in the macaque, vary by 100-fold or so from one area to another, and more areas than in the macaque. A hugely important issue is the general pattern of connectivity. It's a challenge to decipher connectional patterns in humans, and I won't be talking much about that. I'll just lay the anatomical ground truth from studies in Henry Kennedy's lab about the macaque monkey using retrograde tracers. There's basically a 29 by 91 cortical interareal connectivity matrix that's schematized on the lower right, and that has evidence, direct evidence for over 1,600 pathways within that subset. So we can anticipate that the macaque has at least 5,000 interareal connections varying by 5 orders of magnitude in connection strength, and surely our brains are even more complex, but we don't have good numbers yet. Let me come back to this issue of the pattern of convolutions. I think, as all of you know, but is explicitly represented here, here are right hemisphere lateral views of cortical mid-thickness surfaces from 4 individual subjects to show you the diversity of folding patterns. And if you look at inflated versions on the right of these same 4 individuals, one sees that there are some regions like the central sulcus and the superior temporal sulcus that are consistently identifiable in all individuals, but in many regions such as lateral inferotemporal and posterior inferotemporal cortex, the folding patterns are strikingly different from one individual to another. And that's the case even when the same genetic instruction kit is given by looking at monozygotic twins. So here are, in fact, 2 pairs of identical twins. I would challenge you to tell me on even with close scrutiny, which are the twin pairs. I'll just cut to the chase and tell you these are 2 twin pairs, this is another, and look at the diversity of folding patterns despite the commonality of the genetic instructions. So there is some heritability, but it's dominated by variability that arises in early prenatal and postnatal maturation, as we'll come back to. So general questions that we want to explore. Why is the cortex a sheet of tissue, whereas other structures are more blob-like? What determines how much cortex we have, and why and how the cortex fold, and why is it so variable in humans, and how does the cortical parcellation occur? We'll touch upon those. We don't have answers for most of the questions, but we have a context for thinking hard about them. Another hugely important question is how the specificity of connections are established, and that's something that is outside our scope to talk about, just to mention that it's on our wish list of things we could address. So morphogenesis, that is the establishment of the shape of cortical and subcortical structures is driven by, in general terms, how much cells proliferate, where they migrate, how they differentiate, and morphogenesis arises from the physical forces of tension and pressure operating in an organized, coordinated way, as Darcy Thompson articulated more than almost a century ago. If we look at early stages of human cortical development, we see that it really is a very smooth lissencephalic sheet that is a thin shell surrounding a very large ventricular space. And from cellular architectonic studies, we know that a striking feature of early cortical development is that as the cortical plate is formed by migration from proliferating cells deeper down, that the first thing that happens is that cortical pyramidal neurons establish long apical dendrites that are the dominant cellular characteristic. And that's significant, I believe, because a corollary observation is that, at least in tissue culture, but I strongly suspect in the developing brain, neuronal processes, that is axons and dendrites, generate mechanical tension. And why is that important? Well, the hypothesis is that structures that have radial anisotropies, such as that imposed by apical dendrites, if those processes are generating tension, they will tend to tug gently and keep the cortex, even as it grows, keep it a thin sheet. Whereas subcortical structures, such as the thalamus and basal ganglia, tend to have more isotropic cellular elements, and they tend to expand more as blob-like structures. So that's a hypothesis for saying how cellular architecture can play a strong putative role in key aspects of morphogenesis. With regard to the question of how within the cortical sheet cortical areas are generated, that is a intriguing and still unresolved set of observations. One set of relevant points is that a proto-map that establishes the lay of the land for cortical areal specialization appears to occur very early in development, and it appears to be associated with gradients of morphogens within the cortical plate itself, as well as in the ventricular zone. And a number of molecular markers, gene expression patterns, have been identified in recent years that have these morphogenetic gradients and are suspected to play a role in areal specialization. And that's true in the intensively studied mouse, but also in the developing human cortex, especially through the efforts of the BrainSpan project that has studied gene expression patterns from as early as 15 weeks until much later, and showing, again, as in the mouse, broad gradients of morphogens without obvious sharp boundaries to identify candidate distinct areas. So an unresolved, fascinating challenge is how these various molecular markers give rise to differentiation of a highly granular cortical areal mosaic. Are there additional morphogens that just haven't been found yet that are more sharply delineated? Are there area-specific markers that are perhaps related to micro-mRNAs or other molecular species that are only currently being studied? Is neural activity important in this? Probably so, but exactly how these play out is work to be resolved. And why do we have more cortical areas than in monkeys, and far more cortical areas than in mice? There's evidence from mouse model studies by Elizabeth Grove and others that altered expression of specific genes, such as FGF8, I believe it is, can lead to duplication of areas and potential divergence. So there's a framework for thinking about this, but it's largely an unresolved set of issues. So let us turn our attention now to the, going back to the issue of cortical folding patterns. As already noted, the earliest stages of cortical sheet formation give rise to a lissencephalic, smooth cortical sheet for instance, at 25 weeks, just entering into the third trimester. And in that third trimester, a whole lot of cortical folding goes on, and that correlates with the emergence of long-distance connections through the underlying white matter. So that correlation of facts gives rise to an intriguing speculation that invokes, again, the hypothesis that mechanical tension is an important player in the game. So this is an idea I proposed almost 2 decades ago, and I started thinking about it in the context of area V1 and area V2 have a gyrus in between them in the adult, but not at the earliest stages of development. So as cortical folding occurs and the boundary is established along this gyral ridge between V1 and V2, that's around the same time that the long-distance connections from V1 to V2 and also from V2 back to V1 are established. So perhaps those are correlated mechanistically, and that could occur if the wiring, which is topographically very specific from one patch of V1 to retinotopically matched V2, if that were mediated by mechanical tension in a way highly analogous to what I speculated for cerebral cortical sheet formation, that could also account for the emergence of a gyrus in between strongly connected regions. So schematized here, then gyri form between all sets of strongly connected regions, and a sulcus would essentially be the outcome of a losing battle in that competition. Variability in folding could then be a corollary or consequence of variability in area size and or connectivity patterns. So consistency and variability can be hand-in-hand accounted for, at least in principle, by this one tension-based folding hypothesis. So attractive as it is it true? And that's still an open question. I'd say the jury is still out, and there are alternative mechanisms that have been proposed for cortical folding. One of the older ones that has been supported by several publications contending that buckling can occur, that is, as the cortical sheet expands, it may just tend, the increase in surface area may tend to cause buckling in a way to keep the expanding surface area constrained inside the skull. The skull itself can have a shape that's imposed by external factors within the developing fetus, and that may also have an important fact. A particularly intriguing one, I think, is that within the subventricular zone, there may be regions of differential proliferation as proposed by Creasine et al. And Rilo et al. And the reason for taking this very seriously is that in various species, those that are gyrencephalic, including the ferret, cat, as well as human, have a greatly expanded outer subventricular zone compared to lissencephalic mice and other lissencephalic creatures. And this has strongest experimental evidence in the developing ferret, where the regions that have an expanded outer subventricular zone also have a major proliferation of intermediate radial glial cells that produce neurons as well as glia, and these tend to be concentrated underneath regions that emerge into gyral regions, so that the outer subventricular zone is not uniform in its pattern of proliferation. So the concept that emerges that I find attractive is that differential proliferation may well be a major contributor to the formation of the primary folds, such as the Sylvian fissure would be a particularly prominent one in humans. It's hard for me to imagine that differential proliferation on its own could account for all of the secondary and tertiary folds. So I find it attractive to think of a multiple mechanism pattern of cortical development in which later stages of folding may well be dominated by tension, as I speculated before. Empirically, there's evidence that it exists. It is attractive also because if neurons and axons are generating tension, they will tend, perforce, to minimize the overall wiring length, which is a hugely beneficial corollary of brain development. So multiple mechanisms show up often in biology, particularly in developmental biology, and I think that's a good framework for thinking about this particular issue of human cortical and development, well, development in all species. So that covers the ground in terms of basic mechanisms of cortical morphogenesis. Now I want to turn our attention to another set of intriguing issues relating to what happens after birth. We have a methodological challenge in grappling with these issues because at birth, the in vivo MR imaging methods we have show much lower contrast in terms of distinguishing between cortex and white matter and subcortical structures. The contrast is not nearly as great as in the adult. So in a T1 weighted image, the contrast, the cortex is brighter than the underlying white matter, and there's very little contrast. It's a little bit better contrast in the T2 weighted in late gestational periods, and after birth, the T1 weighted images are still low in contrast. The T2 weighted images early postnatally still show contrast, but that actually reduces with time as myelination gets underway. The pattern of myelination and its occurrence in subcortical white matter has a strong regional pattern, as it has been known for almost a century. A classic study by Flechsig showed that some regions myelinate sooner than others, and that is confirmed by in vivo human neuroimaging. So if we look at two basic time points, term gestation, term birth, as shown in the middle panels here. Here are two exemplar individual neonates born as healthy term infants. And here on the right are two exemplar adults. And you can see that most of the convolutions that are obvious in the adult are also evident in the term-born infants, but it doesn't look quite as mature. It's impressive, though, that it's as far along as it is, given that the scaled versions on the left here show that the term-born infants have only about a third the cortical surface area and overall size as in adult. So during these progression postnatally, we can assay, are there regional patterns that are of interest? And if we look at what we call a map of average sulcal depth on the lower right here in term-born infants versus adults, we can see the patterns are similar but not identical. And if we look at a quantitative analysis shown over here, we can see that in terms of the issue of hemispheric asymmetry, the left versus the right hemisphere in an adult shows consistent asymmetries, with particularly the superior temporal sulcus in red being deeper on the right side than on the left. That pattern is also evident, though not quite as prominent, in preterm birth. So one observation is that hemispheric asymmetries are already evident at birth. There are other issues we can look at, though. as shown in this next slide here, starting with the threefold overall cortical expansion between term birth and adulthood. If we look at that pattern systematically across regions, what we find is this map that Carl showed as a highlight in his introductory presentation. So the blue regions here, such as visual cortex and medial temporal cortex, are regions that have expanded postnatally, but only modestly so, about twofold relative to the term control surface area. Whereas these orange and red regions in lateral temporal, parietal, and prefrontal cortex have expanded upwards of fourfold, so twofold more expansion in some regions than others, even though all the neurons are in place, something by virtue of proliferation and migration, something's happening that's very interesting postnatally that we want to explore. And we can get a little bit more insight into the morphometry of this on the lower right here. So in this lateral temporal region that is one of the hotspots of postnatal cortical expansion, we see that in the exemplar folding patterns on the in three representative adults, we can see a deeply convoluted superior temporal sulcus. And in the neonates, we can see a superior temporal sulcus, but it just doesn't look quite as convoluted. So some of this postnatal expansion is by deepening and reshaping of cortical folds. But there's more to be going on that we'll touch upon in a moment. The first, this next slide addresses the timing of this, because the Hill et al. Study I showed you before and replicated here was between adulthood and neonate. The University of North Carolina group, Lee et al., have probed specifically the first and second years after birth compared to term infants, and they've shown that most of the cortical expansion occurs during the first year, a little bit more during the second year, and they found a pattern of cortical expansion during those first 2 years that is colored differently than in the Hill et al. Study, but it shows basically a lot of similarities, hotspots in temporal, parietal, and to some degree prefrontal cortex. There are differences that are intriguing, but it's an open question as to whether those are just methodological confounds because of the exact way in which the analyses were done, or whether there are age-related differences that are neurobiologically real, but that's an open question, as I say. But I think that the next question of interest that puts some valuable context here is looking at the cellular level. So if we look at superficial cells or deep layer cells at different ages, from a newborn to an adulthood, studies by Guy Elston et al. In multiple studies have shown that in prefrontal cortex in particular, that there's a massive elaboration of dendritic arbors that occurs postnatally, and it occurs in a regionally non-uniform way. And another corollary, we'll come back to that, but another corollary of things that change in a regional way are the myelin patterns that I touched upon before, and you'll hear more about from Matt Glasser, but just to give you a quick illustration, in a structural image like T1 weighted image, there are some regions that are brighter than, and some regions that are darker in average in the cortical ribbon. In a T2 weighted image, it tends to be the opposite. There are some regions brighter versus darker. Matt Glasser's idea was to take the ratio voxel by voxel and colorize it, and to use those to generate maps of estimated cortical myelin content. And the hot spots, the red and yellow of heavy myelination, correlate with the pattern of postnatal maturation reported by Flechsig and shown on his schematic map. And the reason for bringing that up here is that there's a correlation also amongst multiple variables. So the regions of high postnatal expansion that we saw before also correlate with regions of evolutionary expansion when we compare human to macaque cortex. These are the hotspots of evolutionary expansion, and these hotspots of developmental and evolutionary expansion tend to correlate with regions of low myelin content, the blue and indigo seas of low myelin content in human cortex. They also tend to be thicker, and as I indicated before, these are also regions that have expanded dendritic arbors, and that expansion occurs mainly during postnatal development. And lastly, one more factoid, if I can get it to advance. There we go. Other studies from John Koss's lab show that in macaque and some evidence in humans that the regions of these hotspots of expansion and low myelin content are also regions of low neuronal cell density. So a number of intriguing factors correlate with one another. And in the last few moments, I want to just touch briefly on the issue of premature infants. If you look at exemplar premature infants, some of them show pathologically underdeveloped cortex, particularly in lateral temporal cortex. And if we look at that systematically, there are regional patterns that are consistent across term equivalent premature infants versus term control. So they're scanned at the same age range, but the premature infants have significant problems that are morphologically most evident in lateral temporal cortex, but turn out to be also evident in other regions as well. So from a study recently published by Yuning Zhang et al., of particular interest are that in medial cortex, prefrontal and cingulate cortex, there are group differences in which the cortex is actually expanded in relative surface area in very preterm infants, and it has a more convoluted pattern. So this is an indication that preterm birth is not simply a developmental delay, but has a more intriguing pattern that is worthy of further exploration. So to summarize, rapid prenatal development is a fascinating study of exploration of its own. Postnatal maturation occurs in a delayed way, but particularly in the first 2 postnatal years. There is regional sensitivity to perturbation and injury, and we can get insights from looking at these issues into the nature of individual variability and the role of experience in shaping cortical circuits. So brain function and connectivity during development is an extremely important issue. It's going to get further valuable evidence from the NIH and other funding agencies are pushing forward with the Human Connectome Project improvements in data acquisition that have already informed us about healthy young adult brain circuits, but through the lifespan RFAs that have just been entering a competition phase and looking at brain development and maturation at different age ranges, and also the Developing Human Connectome Project from the British group are going to give us a huge amount of information. So if I can get this to advance, I want to just acknowledge a number of people in my lab, the group of Terry Inder and Jeff Hill, and several students along the way, and the Human Connectome Project team that's also contributed to some of this story. So thank you for your attention. I'd be happy to answer a couple of questions.
For questions? You showed the lateral surface where those areas expanded. I was wondering, inside of some of those soul sites, say, for example, in the Sylvian fissure, and you think about Wernicke's area, I mean, I would think that that would have expanded in the human. I'm just wondering.
Well, if I understood the question, you're asking, do we have more insight about language-specific maturation during postnatal development?
Yeah, or just the expansion was, you showed us a lateral surface, and so wasn't clear what's going on in those major sulci.
Within the sulci themselves. I would say at the current time, we only have a coarse macroscopic assessment of what's going on. But one of the reasons I'm enthusiastic about these future projects that NIH is planning to support with regard to development and maturation at different stages, that's going to give us insights not only about morphometry, but about connectivity and function through multimodal analyses. So I expect there will be a lot more that can and will be learned in the years to come through such efforts.
No more questions? Okay. Thanks, David. And we can